Hey, Peter here with my FSHD. You know, some of you get your reports back and it will say you have a chromosome 4AL. And you're like, what is that? You know, what's 4AL or what's 4A? You know, it's just, well, anyway, take 2 minutes and we'll go over it for you right now.
All right, so one of the first experimental results you get on your report is here in section 2. And this is your genotyping result and determines whether or not you have a FSHD permissive genetics. You'll remember that you need to have at least one FSHD permissive chromosome 4A in order to develop FSHD. Now, having FSHD permissive genetics does not mean you have FSHD, it just means you have one of the genetic requirements to develop FSHD. So you'll see this individual is 4AL and 4B. 4AL is FSHD permissive, just like 4A, and 4B is non-permissive. Okay, so this individual has at least one FSHD permissive chromosome, so we'll go on and do the methylation analysis. But what is 4AL? Okay, well, first off, let's talk about what's 4A. So 4A is illustrated here. This is the distal most D4Z4 repeat unit. It contains the DUX4 gene that causes FSHD. And you can see the sequence here, the protein coding sequence for the mRNA, exon 2, and then exon 3 has the polyadenylation signal, which is what actually makes this permissive, so that this mRNA can be stabilized and translated out in the cytoplasm to DUX4 protein and lead to FSHD pathology. So 4AL basically makes the same DUX4 mRNA and the same DUX4 protein, but intron 2 here is slightly longer, thus the L. Okay, so intron 2 has this little bit of sequence here, and 4AL has this longer sequence. Okay, why does this matter? Well, because our methylation assay here actually goes from exon 2 to exon 3 and on 4A chromosomes, and so it actually doesn't work on a 4AL chromosome. And so we actually change the assay and we do this region right here, and so we get slightly different numbers because we're doing a different region. So that's why we need to know whether you're 4A or 4AL. So what do these numbers look like? Well, it turns out after doing many assays, we have learned that for reasons we do not understand, the 4AL chromosome in FSHD tends to have higher levels of methylation than 4A. So it's not really comparable because severity doesn't appear to be different. And you know, the same size deletion will just have higher methylation in FSHD1 linked to chromosome 4AL. And so we've changed the metrics. So if you have a 4AL, we're going to look at the first quartile always, okay, and look for it. Typically it's around 30%, but often we see some higher, even into almost to 40%. The key is we've learned that the low range of methylation is actually the most important number. You'll remember that we get the methylation numbers from, you know, anywhere from 20 to 30 chromosomes, and so the range from 6.8 to 89% here. And you get these different quartiles. So these are much higher than you typically see for a 4A chromosome associated with FSHD, but this is kind of fairly typical of what we see for 4AL. Now, those of you who have received reports probably since August of 2021, we've changed our technology to a next generation sequencing technology, and so we get now thousands and thousands of reads of chromosomes, and so it kind of changes the metrics again a little bit. We still get the same kind of range, but the range is from the 5th percentile to the 95th percentile. That's really where we're reading. But what we found is that when we're looking at this, everybody who has confirmed FSHD1 linked to a 4AL chromosome always has several reads that read 0%, totally unmethylated chromosome 4ALs. And we never ever see this in individuals that are healthy or do not have FSHD linked to 4AL. And so this turns out to be a really good new metric to look at, and so we've added it on here. So when we're assaying 4AL, it's really the combination of the first quartile, the low range, and the absolute 0.
Well, there you have it. 4AL is just a slight sequence variation on 4A. It's FSHD permissive. And, you know, it's really important when we do the analysis, and it has slightly different metrics for methylation and determining FSHD. So hope you understand it. If not, send me an email. We'll set up a call, whatever, and we'll make sure that it's all clear. Take care.